And so the project that our group has came up with, which is a group made up of industry, academia, and regulatory representatives, is this categorization of manufacturing components in cellular agriculture. It's a white paper or a published review paper that we are in the process of completing and hope to have published sometime later in the year. The reason that this is of interest to us is, as a research scientist, my goal as a government research scientist is to support the risk assessors by coming up with new methods to help them develop develop ways of assessing if a product is safe, or filling knowledge gaps, which is where something like this comes in. If there's something about a product safety that the regulators aren't quite sure about, they often turn to research scientists such as me to come up with a way of figuring it out. And also just to inform the evaluators and the regulators of the science behind some of the processes so they can better understand how the process works so they can make their decisions in a more informed basis. And that's where another place where a paper like this will come in. So as I mentioned, this project that we have is about medium components. So for anybody out there who's done any cell culture, if you did cell culture maybe 40 years ago, you would have known probably exactly what was going into your culture medium because you had to make it yourself with all of the different salts, all the different vitamins, and all the different other components, which was very difficult. Fortunately, there are companies now like Gibco who sell you these little red bottles. Often you'll have a particular line of cells that you know grows best in, for instance, Ham's F12 medium, and so that's the kind that you buy, and that's about as much as you think about it. But how will this relate to what's going on in a bioreactor where you're producing a cellular agriculture product? What effect will that have? Will it end up in the final product? Is it safe to eat? What are the components? And if we can let regulators know what things are in medium and why they're there, with this graph on the side, which also might be a bit difficult to read at the back, but everything is in there for a reason. And if regulators understand why those things are there and what equivalences there are between other components, it might help them make their decisions a little bit easier. So just in the interest of time, I'll just sort of speed through into our project. So what we did is we took a model cell culture, cellular agriculture culture medium, which was based on a culture medium called Beefy9, which Dave 1 is very familiar with. As you can see, well, maybe you can't see, but he's the lead author on the paper, or the corresponding author on this paper where they defined this medium. And so Beefy9 is a mixture of Ham's F12 and DMEM to commercially available basal media, plus some factors that are required to replace FBS. In the cellular agriculture world, it's not likely that companies are going to want to use FBS, or fetal bovine serum, in their culture medium, because A, it sort of defeats the purpose of it being an animal-free or an animal minimal process. And also, FBS comes with a lot of uncertainties. There could be infectious material in the FBS, and each FBS lot varies from the next, so you don't get the consistency that you might need. So likely, most companies, when they produce their culture medium, are going to use a serum substitute. And this paper sort of boiled that down to the addition of a small number of factors that could replace the activity of fetal bovine serum in their culture medium. And then on top of that, there's things that often that you would normally find in foods. So there's vitamins, there are inorganic salts, which are sometimes involved in foods, and building blocks to build biomolecules. So you'll have glucose, you'll have nucleic acids, you'll have free amino acids, you'll have some fatty acids that are required to help build parts of the cell. And each one of them that we can take a look at individually and make somewhat of a statement on, has that particular ingredient been used in food before? Do we know much about the hazards of consuming that food? What are the, in the toxicology world, you have parameters such as the lowest observable adverse effect limit. So you'll have from either a mouse study or a human study, what is the smallest amount of this product that you can have before you start seeing adverse effects. Or some companies will introduce, some governments will introduce what they call the upper tolerable limit. So basically the maximum amount that they recommend that an individual consumes over the course of one day. And a lot of these ingredients have those parameters already defined for them. And it would be useful sort of to have, all of these in one place in somewhat of a database where you can say, a regulator can look at this paper and say, well, we know that the amount of riboflavin that a person can consume in one day is 50 times the amount that's found in a full liter of culture medium, and so it's not going to be something that is necessarily going to be a risk. So what we're doing with this triage exercise is that we're taking, as I mentioned, each component of BF9 medium and assessing each one individually for its safety based on the parameters that I mentioned. And then once we have this all completed, we're going to assign each of these a risk tier. And this was an idea that was taken from the Upside submission to the FDA. This is sort of the exact thing that they had done. And we thought that was a good way of sort of organizing these into things that need attention and things that we already know are safe or are likely safe. So we have the different risk tiers. We have ingredients that are already approved for use in food. Those ones are very easy. We have ingredients without a history of use in food but are likely safe to consume. And then we have the sort of the gray area. And I think that to me as a research scientist, those are the ingredients that are the most interesting, the things that we don't have the data for, the things that we're not entirely sure what the effects of consuming them would be. Even though they may not end up in the final product, even though they may not adulterate the final meat product, we should still know the hazards involved. And then in tier 4, these are the things I'm not going to mention much further, but these are things like phenol red, where which they know are a chemical hazard that you don't want showing up in the final product. And there are ways of getting around it, like for instance, measuring pH using a sensor instead of phenol red. Just very quickly, in the interest of time. So just some examples. A good example of a tier 1 example would be like niacinamide, vitamin B3. It's approved for use of food. The upper tolerable limit of niacinamide is contained in about 17.5 liters of DMMF12 medium. So it's it would a daily portion of cellular agriculture meat would have to take up 17.5 liters worth of niacinamide to reach the upper tolerable limit, which is unlikely. So it is a lesser hazard. And then you have things like hypoxanthine. So hypoxanthine is a purine nucleobase. So it's integrated into nucleic acids. It's naturally occurring. It's often found in transfer RNAs. It's used in cell culture medium as a source material for nucleic acid synthesis by something called the salvage pathway. So they build new purine nucleic acids by breaking this down and building it back up into other nucleic acids. It does come with some hazard, which we don't know if that hazard actually translates to real risk, but the metabolism of hypoxanthine generates reactive oxygen species. And it's also known that diets high in hypoxanthine are also associated with a greater chance of developing gout in people who are susceptible to gout. So that is the purine that is most likely to be broken down into uric acid. Uric acid crystals end up being one of the physiological causes of gout. So it's unlikely that the cell culture cells would take up enough hypoxanthine to cause a problem, but it's something that should be monitored. So we could give that a tier 2. And then something like a tier 3 example would be insulin. And I should say, actually, these are examples that I'm giving of these tiers, but these are just right now my opinion. As a group, once we have finished putting this together, our goal was to then sort of debate where each of these fit in, because there might be some people who argue that insulin is tier 2. Some people might say it's tier 3. This is just for example purposes, because this is a work in progress. Insulin is something that we don't know exactly the effects of consuming by food a large amount of insulin for somebody who is not, has normal levels of blood glucose, for instance. So there's a higher concentration of insulin found in culture medium than there is in milk. So milk would be an example of someplace where you would actually be consuming insulin already. But the upper tolerable limits of how much per day have never really been identified. On the other hand, it's a lot less than you would be exposed to if you were injecting yourself with a unit of insulin of somebody who has diabetes. So we know that level of insulin is safe for somebody who needs it. But these are questions that we would have to answer as a research community, in order to guarantee that these products are safe. All of that, either these products or these components are not getting into the final product, or we know that there's there's no reason for worry. So this exercise will will accomplish hopefully a number of goals. One, it might act as an example for companies when they're submitting their safety profiles to regulatory agencies. They can do something similar where they triage and tier their components. It can act as a reference point for evaluators where you kind of have a list of all of the toxicological and dietary reference levels of the media components all integrated into one document for quick reference. And I like to use the word demystification for evaluators, so this will help to demystify the science fiction that has been mentioned earlier about cellular agriculture. If you can understand what goes into culture medium and understand why it's there, it becomes less of a magical futuristic thing and a more of a regular manufacturing process. For people like me, it helps to identify data and knowledge gaps, the things that we don't know about safety and we don't know about toxicology. And we hope that it can act as a bit of an example for a future database or living document where obviously the formulations of media from different companies will vary from this. They could be exactly the same. They could be different. We don't know the answer to that unless you've viewed everybody's media component, which we don't, because of trade secrets and intellectual property. This becomes a little bit difficult. So it'd be something that if we could have a database or a living document where either as submissions come in or people or companies can potentially also submit to this on a voluntary basis, and something that we would, I know ILSI has a long history of being able to produce these databases which can maintain privacy, maintain intellectual property, and only be available to viewing by a small group of evaluators and regulators who could then use this information to understand what's coming down the pipeline so they can help prepare themselves for the submissions that are coming, especially if a number of companies that are producing these products continues to grow and grow. Regulatory agencies are going to have to be prepared about what they're going to be looking at if they want to make the decisions in a timely and safe manner. So that's that. Thank you for your time listening, and I can take any questions if we have time. I don't know if we do.
We've got a bit of time for one or two questions.
Yeah, so maybe I'll start. Stéphane Vidry from ILSI Global. So I'm working indeed with Dave and with Melanie on this working group that ILSI US and Canada is leading. So we have a nice representation from academia. We have Guelph, we have Purdue, we have Tufts, we have 3 or so industry, Ajinomoto, Vo, and of course Upside Food. So I think we have a very nice balanced group, and I wanted to thank you again for leading this activity. We're really looking forward to the publication of this white paper. I think the GIFSEN meeting webinars we had a few days ago with Katrina indeed was very successful, and I think there's a lot of hope to see this paper soon, because I think it brings a lot of understanding. So we can take questions now.
This is Tin from ILSI Taiwan again. Thank you for bringing this up. They have a compound quickly get into my mind. It's like a carnitine. Okay, because carnitine has been known can be transferred to the TMAO and cause the cardiovascular problem. So it actually is not the ingredient you put into the cultivation. Actually, it's the cell itself can be accumulated that kind of the metabolite, and because so many of the compound is inside those products, so it's almost impossible to examine every one of them. So this is a good. Thank you for bringing this good exercise, so we can think over what kind of metabolite can be inside those products. Thank you.
Yeah, I guess just touching on that, when we regulate these products, we regulate what's in the final product, not necessarily what's in the medium. If the components of the medium aren't integrated in any way into the final product, they're a process aid. But you're right, then it gives us a chance to look and see what are the metabolites. If we know what's there to begin with, it helps us figure out what will be there at the end. And I think that that's sort of part of the value of this is then regulators can think about, what contaminants might be in the final product rather than just having to start from a blank sheet.
Alan Bubis, Imperial College London. I mean, I also think it's a really welcome exercise. It's going to be really valuable, I think, going forward. And just to get to a bit technical, has the group considered the possibility that at least for some of the components, the bioavailability is different from the bioavailability that the tox reference values were set on, which is often a bugbear for some of the vitamins and minerals?
No, that's something we need. I mean, it would depend on the type of cell, obviously. So each product would probably be different, but it's certainly something that we would have to consider. I think in speaking with some of the people from companies when I talking about this project with them, they say that they know that each one of their individual products, the initial values are going to vary by huge amounts depending on how cells metabolize various things. So. That's why this is just sort of a starting point and maybe more of an example than anything else that people are going to be actually using as a reference document. But yeah, your point is well taken.
Last question over here.
Yes, I want to know if you, as a group, are thinking to consider to think about the nutrient profile of the final products, because we know that meat itself has sodium and another one. So in terms of safety, it's not about toxics or something like that, but we have to think about another things like sodium, for example, for the final products. Do you think about it?
That's something that we could think about as a further project. But I know regulators, if you look at some of these documents that have been submitted to regulatory agencies, they have very charts of each of the down to the the ions and the amino acid profiles, and so
fatty acids, for example.
Yeah. So those all go into the submissions if a country in a particular jurisdiction will ask for it. But that's something we're our group is looking always for further projects, and that could be something that we could look at.
From a company perspective, that is something that we worry about. If there's high cholesterol, for example, even though, cholesterol is not something we, from a safety perspective that we worry about, but it's really more the consumer perception. If something's high in cholesterol, why would you eat that? So, but thank you everyone. Do enjoy the rest of the meeting.